So I think I'm going to exactly connect to what Ludovic has just presented right now, and I'm going to go into a little bit more details on FDR control and set this a little bit more into context. And these are very recent things that we evaluated in our lab. So this is really like the first part that I'm going to present has just been accepted and is in press right now. The second thing I'm going to talk about is a cross-run alignment to boost confidence and to increase the completeness of data matrices. Exactly. So to begin with, I'd like to talk about peptide-centric scoring in context, and I'm going to come to what I mean with in context in a minute. Just as a quick recap of what Ludovic has been talking about in the last hour. So the main 3 steps that the peptide-centric analysis include are, first of all, automated chromatogram extraction and peak group scoring, where you extract the scores for each target and decoy in the spectral library. And then in the next step, you perform the semi-supervised learning to calculate the discriminant score that best separates your targets from the decoys. And then based on these discriminant scores, you can do statistical scoring based on the Story-Cipriani approach to then get Q values, which are corrected for multiple testing. However, when you look at this workflow, there are 2 things that probably come up. One thing is that you do the error estimation on the peak group level or peptide query level only. The second thing is that you do this targeted extraction and statistical scoring on 1 sample at a time. But if you think of your experiments in your lab, most studies report peptides and proteins and not actually the peak groups or peptide queries. And the other thing is that most studies include more than just 1 sample. So the question is, does this actually make a difference? And the answer is it does make a difference depending on your experimental setup and the way that you analyze your data. So the first thing that is important is the size and comprehensiveness of the spectral library. And this is something that Mike was already touching on. And that was a question here from Timo, I think, and I'll talk about this. The second thing is the number and heterogeneity of the studied proteomics samples. And most importantly, there's the relationship between those 2, so the proteins present in your samples and the protein set that is represented in your spectral library. So when we're talking about this spectral library, historically, at least in our lab, the target DAA analysis developed from this SRM perspective. And here you usually build a library based on like your specific sample, and this is fairly well defined. So you have a sample set which is fairly homogeneous. You, for example, do DDA measurements and build a sample-specific library, and then perform a really targeted analysis of the DIA measurements. However, this type of analysis is not always possible, or you don't always want to do it, and you can scale this approach up to more comprehensive spectral libraries. This is already the case, as what Timo suggested. You have quite a heterogeneous data set, and you want to build a library, then you cannot call this anymore really sample-specific, because all the samples are very different. So now you're maybe having something like a study-specific library that is already quite large and diverse, and you analyze your data in a probably a little bit less targeted way. Another option is that you actually use quite a large compendium of different samples to build your library, or you can take a repository that has been published before, and I think Tina mentioned a few of those yesterday, and then you have such a repository scale library. And then I'm not sure if you can still talk about targeted proteomics, but then you're basically in a more untargeted fashion extracting these peptides from your data. And this has very important, or this results in very important considerations for the FDR control. And the most important thing is here, when we now look at the discriminant score distributions that Ludovic has explained in the previous lecture, they look very, very different depending on what kind of library you use. So if you have a sample-specific library, then the majority of your peptides and also proteins are detectable in your samples. So when you look at this discriminant score distribution, you see here in gray this time the decoys, and in red the targets. So you have a very, very small fraction of targets that fall in this decoy distribution. On the other hand, you have this comprehensive library and you analyze the data set, then you see that your fraction of detectable targets is very small and you have a large fraction that is actually overlapping with the decoys. And this has a specific term, which Mike just mentioned, which is this pi0. And this is just the fraction of false or non-detectable targets and can be calculated or estimated as the number of non-detectable targets over all targets in your spectral library. And the big thing is that you have to keep in mind is that the larger this pi0 is, the higher is your statistical burden. And this has several effects that need to be considered. So one thing is that if you have a large fraction of false targets, then your peptide query FDR and your protein level FDR start to diverge. So in this schematic plot here, you can see, okay, we have on the x-axis the peptide query FDR. Let's say we go to 1%. We go up here. This is the match to the protein level FDR. And you can see, oh, oops, we have 5% protein level FDR. So the question is, what's going on here? So probably let's take a toy example. So here we have detected in our data set a bunch of peptides, and the green ones are the true positives, and we allowed 15% peptide level FDR, and so we have 3 false positives in our detected peptide list. If we now want to report proteins, what's happening is that the true peptides tend to group together to map to one true positive protein. However, the decoy detected decoys are quite random. So each of these peptides tend to map to a different protein. So at the end, what you end up with is 3 out of these 7 proteins are actually false positives, and you have an increased protein level FDR compared to your peptide level FDR. And this is something that you need to be specifically aware of if you use such large libraries. And for this, we have now in this, again, new version of PyProfit implemented a protein level-based FDR estimation. And the way that this is done is basically by taking the best scoring peak group per protein to get a surrogate for a protein level discriminant score distribution. And you can see this basically down here. So here's for the peptide queries of one specific sample, the discriminant score distribution. And then we took the best peptide query per protein to derive a protein level discriminant score distribution. Here you can already see that your fraction of false targets has decreased quite a bit from the peptide to the protein level. And then you can, based on this discriminant score distribution, do the Q-value estimation based on the story-tip-Shirani approach that Ludovic explained just before. So using this, you can control the protein level FDR in your samples. So this should be covered. Now, a second effect comes into place if you want to analyze an entire data set, because the effect of error accumulation can take place. So here, for example, if we stay at this 1% peptide query FDR, we have 5% FDR on protein level for 1 run, but if we go to 20 runs, we actually go up to almost 25% global protein FDR. So what can we do about this, and how does this happen? Now here's again like just a small toy case example. Imagine you have a spectral library which contains again these 7 proteins, and then we extract data from 5 different samples, and these are all kind of the same samples, so the true positive proteins that you detect are always the same. However, the decoys again match randomly, and what happens is that the 3 other proteins in your spectral library are randomly hit in one of each of these samples. So while in each of these separate runs you have a 20% protein level FDR, you still, when you report the total number of proteins that were detectable in your entire data set, you suddenly end up at a 42% global protein FDR. And now the question is, do you actually want to report this, or what do you want to report, and how can you correct for this? So thing that we thought of are different questions that you can ask, and depending on the question that you ask and that you want to answer in your specific analysis, you need to perform different FDR estimations. So one question could, for example, be whether a protein is detectable in one specific sample. And then you're really only specifically interested in one sample, and this is what we call the run-specific context. And this is also what's the normal thing that's done in mProfit or PyProfit. And the way that this works is basically when you have a scoring matrix like this one. So here are 3 different runs. These are the different, let's say, proteins. And for each of them, you have a different discriminant score. And then in the run-specific context, you take one run at a time. You look at the discriminant score distribution and transform your d-score into a q-value. And this is then your confidence level for the specific protein A1. If you now have want to analyze a different run, you do this again for each different sample. And what you then realize is that here you have a similar d-score compared to here, but your q-value suddenly is much larger. So how does this happen? This happens when you have a different prevalence of your targets in your sample. So if you want to compare these 3 data sets, or these 3 samples within your data set, the same kind of quality peak groups can have a very different statistical score at the end. And you will probably find this detected here, but you will kick them out in these 2 data sets. So one way that a comparability or a better comparability between runs is possible is with an experiment-wide context. And here you're interested in which subset of samples a protein could be detected in. And then you're interested really in the entire matrix here. And for this, you basically can take the discriminant scores across all the samples in your study. You make a mixture distribution which will have kind of an average pi0, an average fraction of false positives. And then you get comparable q-values across your data set, and these will correspond better to the quality or the visually observed peaks that you have detected. So these do still not cover the problem of error accumulation across the runs. So there's a third question that you could want to answer, which is the detection, whether a protein was detected in at least one of these samples, and you want to report the global number of proteins or the global set of proteins that you could detect. And for this, you're only interested in the best detection across all these samples, and this is what we call the global context. And for this, you can basically take the best scoring the best d-score per analyte and make a discriminant score distribution for this one. You will get an optimal q-value for this one, and you will control the FDR on this global set. And this can be used to generate a master list of detectable proteins. And if you use this, you can make sure that you don't have a large error accumulation across large data sets. And I'll show this in a few slides. So our suggestion is to use this global context to build a global master list, and then use this global analyte constraint to control error accumulation in either one of these 2 contexts. And the practical implementation of this is again another profit, which is what we at least internally call the jumbo PyProfit. PyProfit itself is just a Python-based re-implementation of the mProfit algorithm, including these DIA scores. And what's new in this jumbo PyProfit is that we perform the semi-supervised learning step that Ludovic mentioned, not on a per-run basis, but we subsample across the entire data set and then make a global machine learning to get the best weights for each of these scores for the entire data set so that these scores are better comparable. Then we perform FDR on peptide query, peptide and protein level to make sure that we can deal with large data, with comprehensive spectral libraries. And then the last thing is that we perform a context-dependent FDR estimation and allow you to assess the FDR in a run-specific experiment-wide and global context. And this is available on GitHub in the PyProfit repository. And there's a small tutorial in the appendix of tutorial 4. So this is a rather complicated list of the steps that you have to perform to do this, but we have like a few commands for you to test if you want in the appendix of tutorial 4 on Thursday, I think. And to show you the effect of these different error control strategies, I want to show you one case study which we used in the lab to evaluate all of these, which is the SWATH-MS Interlaboratory study. It's not important in this context to understand exactly the complex setup of the study, but what's important is that the same HEC-293 cell lysate was analyzed across multiple labs, and in total this resulted in 229 DIA measurements of the exact same HEC-293 cell lysate, and this was centrally analyzed with OpenSWATH and the new PyProfit. And the questions that we specifically wanted to ask was, what's the impact of using spectral libraries with varying size and sample representation? And secondly, what are the implications of these different error control strategies that I just presented to you? So the 3 libraries that we used for this are, first of all, the combined human assay library CAL that already came up yesterday in a few lectures. And this basically consists of 331 DDA runs from different cell lines, including HEC-293. And the second library that we used was a HEC-293 subset of these samples that are represented in the combined assay library. And these were only then 134 runs, and the last one is the sample-specific library, which we built from 6 unfractionated HEC-293 samples from this Interlab study. And here you can see that the combined assay library consists roughly 10,000 proteins. The HEC subset then is a subset of this CAL containing roughly 6,000 proteins, and the sample-specific library is fairly small with 2,500 or so proteins included. And if we look at the protein level discriminant score distributions, this is using the global model for this data set. You can see the discriminant score here on the x-axis, and in yellow you can see the discriminant score distribution for the combined assay library, and you can see that there's a large amount of non-detectable false targets. When you go to a more sample close library, which is the HEC library, then your fraction of false targets gets a little bit smaller. And if you use the sample-specific library, you basically don't have any false targets, and all your targets are detectable. So the effect that this has on the global analysis of this data set is quite drastic. So if we use the combined assay library and we do the FDR estimation only on the peptide query level, as is done in like standard and PyProfit, then you can see that on protein level, the number of proteins that are cumulatively detected across this 230 runs is increasing quite drastically, although we are analyzing the exact same data set over and over again, right? So this is not, this you shouldn't see. If we then say, okay, we control the error on the protein level, you see that you're getting much fewer protein detections, but you still accumulate detections across runs. And if we set the global protein FDR threshold, then you see we can find a consistent set of protein targets and avoid error accumulation across this large data set. And it's now very interesting to compare this to the 2 other libraries. So here in pink are the data points for the HEC subset library, and you can see that there's a much smaller difference between the different error control strategies, but you can still see accumulation of targets or false targets with increasing data set size. And if you use the sample-specific library, it basically doesn't matter what kind of error control you do, because all of your targets are anyway detected in your sample, and you have no problems with error accumulation whatsoever. So if you look at the total number of detected proteins, you can see that for both the combined assay library and the HEC subset library, you find almost the exact same amount of proteins. these few proteins that are exclusively found with the combined assay library represents kind of the opportunity gained due to a larger search space and a few things that were probably too low abundant in the HEC cells to be detected in DDA. The HEC library also has a few unique detections, which comes from which is most likely coming from the fact that the statistical testing burden for the combined assay library is much, much larger. So these are things that you can find because you have to less correct for multiple testing, right? So the scaling that you have to do, or that you are allowed to do, is much larger, so you can shift your FDR threshold to the right, as Mike and Ludovic have already pointed out. And the last library is the sample-specific one, and here you can see, well, you're doing great in terms of you identified 100% of the library, but in general, you are losing quite or missing quite a lot of the proteins because they were just not present in your library. So this is just a very short other example where we tested this. This is a more realistic data set of human blood plasma cohort, which was published in 2005 by Yang Sheng. And this includes 246 blood plasma samples from 116 individuals. And we've also analyzed this with two different libraries. One's the combined assay library, which definitely doesn't include so many, or like is much, much bigger than what you could ever detect in a single plasma sample. And we also generated a plasma subset from the few samples that were included in the combined assay library that were coming from plasma. And here you can see the very differently looking distribution of the discriminant scores. So for the plasma library, you are able to detect a fair amount of the proteins in this library, but for the combined assay library, only a very, very, very tiny fraction of the library is actually represented in the samples. And now the nice thing is, if you do the error control quite properly across the entire data set, you get a global set of proteins that is comparable between those two. Again, the CAL has more opportunities to find something, while the plasma sample doesn't have to correct for that much multiple testing. So just to again make you aware that this error control on the global level is extremely important. When you don't do this and you do the normal standard mProfit, PyProfit with this super large library that is way too big for plasma, you're accumulating proteins across your data set, claiming at the end probably 3,000 proteins in your plasma sample, which is unrealistic, and there's a definite need to make sure that you control the error on the protein level and across the data set. So what we've learned, going back to the questions that I asked at the beginning, what is the impact of using different size and representation spectral libraries? There's definitely a trade-off between comprehensiveness and multiple testing burden, and you need to be aware that when you use large libraries, you can benefit from the opportunity that is gained by having a larger set of proteins available in this library. However, you have to test for multiple testing, and it's very important to do the error control properly. And this already leads to the second question, what are the implications of these different error control strategies? And so from, at least in our hands, we saw that for sample-specific libraries, the error control on the peptide query level and without considering the entire study should be generally sufficient. However, the larger the library and the more comprehensive, it is absolutely essential to do the error control on all analyte levels up to the protein level, and also to consider the context of your study and not only control the error on one single sample. So now the question is, of course, okay, which library should I use then? This is a very good question, and there's, of course, I think there's still a lot of discussion going on in the field how to best solve this problem. Generally, what I would say is that a sample-specific library is totally viable if you have very specific samples and all your samples are quite similar to each other, so you have a specific cell line and a specific question that you're super interested in. However, if you have then like a heterogeneous sample cohort, you're automatically, even if you don't necessarily intend to, you have a more comprehensive library, and at this point you have to be aware of this error accumulation and to do the error control properly. The next thing is, if you don't want to go through the hassle of library generation, and you've seen that this can be quite a pain yesterday in the tutorials, and you want to use a library resource, then it's very important to see that you know how well your sample is represented in this library, and if it's well represented, you still have to see that you do the error control properly. And if your sample is not well represented in this library, don't use it, because you can only lose from having a too large search space with very little things to actually find. With this, I finish up the first part, and now finally get to the second part of this presentation, which will be on the transfer of peptide detection confidence across samples in the software TRIC. And this is a very nice software that was developed by Hannes Rist and that has been published last year. And this is one thing that is still quite unique to our analysis pipeline, and it's it's nice because it increases the data completeness and consistency of detection across a large data set. And the way that it does it is by using the information across all runs to boost the confidence of detection in single runs for certain peptide peak groups. And this is done by doing a nonlinear alignment of between the different samples. And so here you can see, well, you have this peak group in this specific run, and this might not match to the exact retention time in the second run, but so here it basically looks linear, but it's done in a nonlinear fashion, and you can you can match these peak groups together, and you can also find runs where the peak group is actually not being detected. So just to go through in a little bit more detail through the methodology. So what is done in the different steps is, first of all, you compute an optimal guidance tree, and this is done by doing a run-to-run pairwise distance matrix to basically define which runs are close to each other in retention time space. Once you have built this tree based on this retention time closeness, you can compute the local regression, and this is done in a non-linear way. And this is not dependent on your IRT peptides, but this is really done on all the peptide identifications that you share between any two runs. And once you've done this, you can select a high quality or the highest quality starting point for each peak group independently. So you start from your best peak group for this peak group in the best run that you found, and then you travel through this tree and align and then transfer the confidence across these runs. So this means that if you have very high confidence here, you have very high confidence detection here, but your confidence has actually not been very high here, this confidence is boosted, and you get this, you recover this peak group for your final data matrix. The next thing that you can do is you can revisit nodes where you didn't find a peak group, right? So here you can imagine here you don't have this peptide peak group detected, and you can go back there and integrate the noise at the specific retention time that has been aligned, and this can be then used as a background noise to be able to calculate fold changes at the end and to have a complete matrix. This needs to be taken a little bit carefully, because this is assuming that your peptide is below the limit of detection and that there's nothing else in this region. However, if you have now like a bad interference in this region, you might you're integrating noise unselectively, right? So when you have a bad interference in this region, then you might get super high values. So this step is really something that you need to be quite careful about, and I would suggest to manually inspect this again if you want to use requantification. But in general, if you look at the performance of this TRIC alignment, you can see here on the right that validating this based on 7,000 visually inspected chromatographic peak groups, the TRIC results in a much better ROC curve characteristic than our standard workflow in black. And if you look here, this was validated on a heavy light quantification pulse-Silac experiment. And what you can see is that here on the x-axis and this distribution up here, the heavy light elution time differences that are very similar for the high quality pairs and the aligned pairs, just having a wider distribution for the aligned pairs. And what you can see in terms of intensity, that generally, as you would expect, the realigned pairs were of lower intensity. So what this means for your data and in terms of data completeness is that you can basically have a much more complete data matrix once you use this alignment. And here this was based on a data set from 178 yeast lysates. And here you can see the number of detected proteotypic peptides per run. And there's quite a lot of variation across all of these runs. And the matrix completeness is at like roughly 60% or 80% on peptide and protein level respectively. But if you move and do this, the TRIC alignment in this nonlinear fashion with the guided tree, you can see that you have much more equal detection values, and the completeness increased to 80 or 90% in this data matrix. So you have improved data consistency and a better completeness in this quantification matrix. So with this, I'm already getting to the summary. So I wanted to talk about this peptide-centric scoring in context, and I hope that I could convince you that different questions require different FDR estimations in your data set, and that there is a trade-off between using spectral libraries that are comprehensive and which you then have to correct for multiple testing quite a bit. The important fact here is that if you have such comprehensive libraries, you have large fractions of false targets, and in these cases you have to control for protein level FDR, and you need to look at your data set in global and to avoid accumulation of false positives. And finally, you can use this the TRIC algorithm to increase the consistency and completeness of your data matrices by using this retention time alignment across the runs, and by transferring the confidence of your identifications between the runs. And yeah, now I think you're not sitting as comfortable as him, but maybe you also have some questions, so I'm happy to answer. Yeah.
Great, thanks.
Yeah, so I have a question regarding this TRIC. Which softwares or programs is it nowadays implemented in? Do you know?
So this is a Python implementation.
Okay, so.
So Brendan.
Yeah, well, so we, an intern of mine who unfortunately is heading to Google in 3 weeks, has been working on this for about a year, and I'm excited because it looks like you presented some new validation data, because we've been feeling somewhat limited by the validation data that we have, but we think we're pretty close. We've thought we're pretty close for a little while now, but yeah, we just feel like we need to validate all, it a second time and make sure that it works as well inside Skyline as is reported in the paper. So do you have how many? I wasn't clear on how many new validation sets you presented there that weren't in the paper.
So the only data set that was not in the data is this one, and
okay.
Probably, I don't know, Ludovic.
Well, what was the curated one before?
This is in the paper.
That's the paper one.
That's the paper one, yes.
Okay. 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 Well, anyway, I'll talk to you.
Yeah, I have a question. We haven't done these exercises yet, but are these kind of, I call it also a bit quality control plots, kind of standard outputs that we have in this lab when we execute, let's say, open swath, and where this should be implemented, or how can we access and get this information that is so important for us to check?
So you mean the discriminant score distributions and the p-value histograms?
Yeah, exactly. So that we can get a clue what went wrong, or if we don't necessarily want to directly access the data in some different way.
Yeah. So I mean, what you get is when you in the tutorials, also on Thursday, I think, you will run PyProfit, and what you get is at the end is a PDF which will report the discriminant score distributions and the p-value histograms. And the point where you have to be careful is when you see that that you have this. Let's quickly go back. So as soon as you see that your distribution looks more like the yellow one and not like this red or pink one, then you should think of whether you should not please consider doing a protein level FDR control, and when you want to assess large data sets, that you control for error accumulation across this data set. So I think that it's quite important to assess these discriminant score distributions. The things you should look for is, first of all, this like the distribution of the targets to these two distributions. The second thing is that the false targets should overlap very nicely with the decoys. So as Ludovic said, these have to match. And the next thing is that you can look at the p-value histograms and see that you kind of really level out at some point, and that you don't have like very strange characteristics shown in these histograms. And these are reported from the pipelines, and you can also see them in Skyline, I think.
Do you have maybe some data sets that you are going to show where things have failed and where the precise error was then spotted in the end? So just for
I mean, so the data set where you definitely see it is when you go here, right? So if you analyze the same data set over and over again, and you're accumulating your targets, like the number of reported proteins, almost reaching library level, which is like 10,000 proteins, then you're definitely doing something wrong, right? So it's quite hard to assess this and to see it in a heterogeneous data set, but this was, for this, a nice showcase where you should see a flat line, but we saw accumulation.
May I again ask what is about the minimum library size, which would be useful, because we still need to approximate the decoys distribution, and I mean in such experiments where we have only several proteins, for example in AP or something samples. Good question.
So I don't know if I can give you a good number for the minimum size. I'd say so in general you need a fair amount of proteins or like of peptide queries if you say like just the size of your assay library to be able to look at distributions, right? If you have too few, then you don't have a distribution. I'd say like probably hundreds should be in there, but I think that this is really a question where also opinions diverge a little bit. So I mean, Alexei will, I think, talk tomorrow, and he's generally of the opinion that you should, if you have a data set that has a lot of peptides in there, you should also kind of look for all of them. And then I think what Mike's been saying more is when you have a few peptides you're interested in, it's totally sufficient to look for those. You definitely still have to have enough peptides to make your model, but then you're done, right? So I don't know, I would say maybe 100 or so. Do you agree?
I think it just depends on what you need. But yes, you do need to have enough in order to assess your model. I mean, we've never done it for less than 100. I would say it does more than that. So.
Yeah, and one of the data sets I'm going to be presenting is actually that TRIC data set when we do hands-on tomorrow. and there I do both sort of the proteome-wide, it's a small proteome, as well as 250. So, or no, 450, sorry. So, yeah, I would say hundreds to, yeah, like 100 or to 500 would be fine.
I just want to add one point that as you're considering that question, I mean, there's the issue of how do you get these estimates? But the thing that's often not assessed is when you have an FDR estimate, there is some variance associated with that estimate. And as the size of the data set becomes smaller, the variance increases relative to the FDR. So you might think you're controlling it at a 1% FDR, but it's plus or minus 5%, because your variance is so large. So that's sort of the hidden consideration. It's very difficult to ascertain with these methods because you only have a single fixed set of decoys.
But could we extract another set of decoys or another 10 sets of decoys and look at the stability of that? Is that what you would?
Yes, you can do that. It's not. Yeah, I mean that's more or less the standard way to do it. The question is, how do you get enough decoys in this setting that look different from each other, right? Because of the kind of things that Ludovic talked about, where you're trying to ensure that the decoys resemble the targets, right? Now you try to generate the next set of decoys where they're not totally independent from the first set of decoys. So statistically, it's very difficult to estimate the true underlying variance.